Welcome to the deep dive. Today we're tackling a really foundational topic, the absolutely pivotal role bacteria played, well, in the genesis and evolution of all complex life.
That's right.
You're here because you want to get smart, fast, and trust us. This story has some genuinely mind-blowing turns.
Absolutely. Our mission in this deep dive, is to sort of unravel how these seemingly simple organisms didn't just come first. They fundamentally shaped the very cells that make up everything, from a fungus to, well, to you listening right now.
And we're digging into some incredible scientific research that lays out this epic evolutionary journey. Okay, let's get into it. Where does this whole astonishing story even begin?
Well, it starts billions and billions of years ago on an early Earth that was, well, it was almost alien compared to today. We're talking the Archean eon.
Okay.
Picture this. Constant volcanic eruptions, an atmosphere completely lacking oxygen.
No oxygen at all.
None that we need to breathe. Plus, intense ultraviolet radiation bombarding the surface. Not exactly a welcoming place.
Doesn't sound like it.
But crucially, there was liquid water. And that changes everything.
So this incredibly hostile, oxygen-free world, that was actually the cradle of life.
Exactly. This extreme environment acted as a kind of crucible, a powerful testing ground for abiogenesis, the origin of life from non-living matter.
Right.
The harsh conditions put immense selective pressure on these early cells. It really favored those with metabolisms that could function without oxygen.
Anaerobic life.
Precisely. And it's in this context, we think, that we see the initial split between the two major domains of prokaryotic life, bacteria and archaea.
Ah, okay.
Maybe they quickly specialized, taking advantage of different extreme niches. Like archaea seem to have perhaps gravitated towards high-temperature environments pretty early on.
Interesting. So, okay, what were these earliest life forms actually doing? How did they survive in such a brutal world?
Well, one early and really successful strategy was something called chemolithoautotrophy.
Chemolithoautotrophy. Okay, break that down.
Think of it like this. They could essentially eat rocks, deriving energy from inorganic chemical reactions, and breathe CO2, building their own organic molecules using carbon dioxide as the source.
Oh, eating rocks and breathing CO2.
Kind of. And we actually have evidence for methane-producing bacteria, these things called methanogens, dating back a staggering 3.46 billion years.
3.5 billion years ago.
Yeah. It suggests these kinds of chemical energy strategies were really ancient, probably even predating photosynthesis, so early metabolic abilities likely developed in stages, building up in sophistication.
That's just incredible to wrap your head around. But how can we possibly know what existed that far back? It's not like finding a perfectly preserved fossil, is it?
Well, no, not exactly, but we do have clues. Pinpointing the absolute first flicker of life is definitely a challenge.
I bet.
We have things like carbon isotope readings. From rocks in Greenland, there are hints of biological activity, maybe as far back as 3.8, even 3.85 billion years ago.
Hints, so not definitive.
It's suggestive, but debated. Then we have microfossils, tiny ancient structures that look like cells. Some findings from Greenland around 3.8 billion years are still pretty contentious.
Okay.
But we have more widely accepted bacterial microfossils from Australia. Those are about 3.5 billion years old.
That seems more solid.
It is. And then there are stromatolites. You might have seen pictures, these layered kind of rock-like structures.
Yeah, those wavy formations.
Exactly. Often formed by the activity of microbial mats, particularly cyanobacteria. And these also date back to around 3.5 billion years ago. So that gives us visible macroscopic evidence of bacteria physically changing the planet.
For 3.5 billion years.
Yeah.
It's just mind-boggling.
It really is. So the current scientific understanding is that bacteria have been around since the early Precambrian, maybe 3.5 to 3.8 billion years ago. And bacteria and archaea seem to have diverged very early in that period. But like you said, interpreting these ancient clues isn't simple. It requires a multidisciplinary approach, looking at the morphology, the shape, the chemistry, the isotopes, the surrounding geology, all of it, to build a convincing case.
It sounds like forensic science on a planetary scale.
Yeah.
A very, very cold case.
That's a good way to put it.
And what strikes me is how quickly life seems to have popped up after the Earth cooled enough to actually have liquid water.
Yeah, that's a really key point. The oldest rocks we have are around 4 billion years old, maybe a bit older. And we see these signs of life appearing just a few hundred million years later. Geologically speaking, that's pretty fast.
So what does that speed suggest?
Well, it suggests that abiogenesis, the origin of life itself, might have been a more probable event than we sometimes imagine. Perhaps the conditions were just right.
Hmm.
Of course, there's always the alternative idea, panspermia, that life originated elsewhere and arrived on Earth, maybe on a meteorite.
Right, the extraterrestrial origin idea.
But the current evidence, it really does strongly support a terrestrial beginning, life starting right here.
Okay. And before we had DNA running the show, there was this idea of an RNA world, wasn't there?
Exactly. The RNA world hypothesis is a really important concept here. It proposes that in very early life, RNA, which is chemically similar to DNA, did double duty. It served as both the genetic material, carrying the instructions,
Like DNA does now.
right, and also as the main catalytic molecule doing the work of enzymes.
So information and action in one molecule.
Pretty much. Think of it as a molecular Swiss Army knife. RNA can store information, and it can also catalyze reactions. These catalytic RNAs are called ribozymes.
Okay.
This could have been a crucial intermediate step, eventually leading to the division of labor we see today. DNA mainly stores info, proteins do most of the catalytic work, and this RNA world stage might have also set the scene for that early divergence of bacteria and archaea.
Okay, so we've got these ancient bacterial ancestors, potentially arising from an RNA world. What did their cells actually look like? We often hear prokaryotic and think simple. Is that fair?
You know, that impression of simplicity can be really misleading. While it's true, bacterial cells lack the complex internal membrane-bound compartments, the organelles, that eukaryotic cells have.
It's like a nucleus or mitochondria.
Exactly. They don't have those, but they are incredibly efficient and adaptable. It's a highly refined prokaryotic design honed over billions of years of evolution. Far from simple, really.
Okay, so give us the tour. What are the essential parts of a typical bacterial cell?
All right, let's start from the outside in. You've got the cell envelope. This is the bacterium's interface with its world, a layered system.
Layers of protection.
Protection and interaction, yeah. The innermost layer is the cytoplasmic membrane. It's thin, made of phospholipids and proteins, and it's selectively permeable. It controls what gets in and out.
Like a gatekeeper.
Precisely. Then outside of that, in most bacteria, is the cell wall. This is a more rigid layer, usually made of a unique substance called peptidoglycan.
Peptidoglycan. That sounds important.
It is. It gives the cell its shape and structural support, protects it from osmotic stress, and it anchors things like flagella. Interestingly, though, some bacteria, like mycoplasma, have actually lost their cell wall entirely.
Huh. So there's variation even there.
No, absolutely. And then many bacteria have yet another outer layer called a capsule, or sometimes a slime layer, often made of polysaccharides, sugars.
A sugary coat. What's that for?
It can be quite thick. It helps them stick to surfaces, prevents them from drying out, and importantly, it can help them evade the immune system of a host organism. It can be a major virulence factor, making bacteria more dangerous. The capsule of Bacillus anthracis, the anthrax bacterium, is actually polypeptide-based, but it's a key part of why it's so pathogenic. Though not all capsules are about disease, many are just protective.
Okay, so a multi-layered defense system. What about the inside? What's going on in there?
Inside, you have the cytoplasm. It's this gel-like matrix, mostly water, but packed with everything the cell needs. This is where growth, metabolism, replication, all the action happens.
So enzymes, nutrients.
Enzymes, nutrients, ribosomes, the protein-building machines, and of course the genetic material. But crucially, no membrane-bound organelles floating around.
Right, no internal compartments. Where's the DNA then?
The DNA, typically a single circular chromosome, is located in a region called the nucleoid. But the nucleoid itself isn't enclosed by a membrane. It's just sort of concentrated in one area of the cytoplasm.
Got it. And you mentioned ribosomes.
Yes, the ribosomes. These are the molecular workbenches where proteins get assembled based on the genetic instructions. Bacterial ribosomes are a bit smaller than eukaryotic ones. We call them 70S ribosomes versus the 80S in eukaryotes.
And that difference matters.
It matters hugely for medicine. That subtle size difference is why many antibiotics can target bacterial protein synthesis and kill bacteria without harming our own cells. A very convenient evolutionary quirk for us.
That is clever. What else is in the cytoplasm?
Well, often you find plasmids. These are small, usually circular, extra pieces of DNA. They hang out in the cytoplasm and can replicate independently of the main chromosome.
Extra DNA. What does it do?
Plasmids carry genes that aren't strictly essential for the bacterium's day-to-day survival under normal conditions, but they often carry genes that provide advantages in certain situations, things like antibiotic resistance genes, or genes for virulence factors, or the ability to break down unusual food sources.
Ah, so that's where antibiotic resistance often comes from, those pesky plasmids.
Very often, yes. They're key players in the rapid spread of resistance.
Okay. And how do bacteria move around and interact physically?
They have various appendages sticking out. Flagella are the most famous, long, whip-like structures they use for motility, for swimming around.
Like little propellers.
Exactly. And they're powered in a fascinating way by something called the proton motive force, basically, a flow of protons across the cell membrane, like a tiny electrical current. Not directly by ATP, the cell's main energy currency, which is interesting.
Huh. Okay. Yep.
Then there are pili and fimbriae. These are shorter, more hair-like structures. Fimbriae mainly help bacteria stick to surfaces, kind of like bacterial Velcro. And then there are specialized pili called sex pili, which are involved in conjugation.
Sex pili. For bacterial sex.
Well, sort of. It's not reproduction, but it's a way they directly transfer DNA to each other. We'll definitely talk more about that.
Okay. So even though they're microscopic, they're packed with sophisticated machinery. You mentioned their efficiency. Where does that primarily come from?
A lot of it boils down to their tiny size. We're talking typically 0.1 to maybe 5.00 micrometers across. Really small.
And small is efficient.
In this case, yes. Because they're so small, nutrients and waste products can diffuse rapidly throughout the entire cell. There's no need for complex internal transport systems like in our much larger eukaryotic cells.
Ah, makes sense.
And their high surface area to volume ratio means they have a lot of membrane surface relative to their internal volume. This facilitates rapid exchange of materials with the environment, getting food in, waste out.
Efficient exchange.
Exactly. And that variability, we talked about the capsules, pili, flagella, plasmids, that reflects the incredibly diverse and often harsh selective pressures bacteria face. The genes for these variable features are often on those easily swapped plasmids, allowing for incredibly rapid adaptation to changing environments, nutrient sources, or host defenses.
Amazing. Tiny, efficient, adaptable powerhouses. Now, you mentioned their metabolic diversity earlier. This seems to be where bacteria truly shine, doesn't it?
Oh, absolutely. It's almost unbelievable. Bacteria possess an unparalleled range of metabolic capabilities. It's what allows them to thrive in practically every habitat on Earth, from boiling hot springs to Antarctic ice, and why they're so crucial for the planet's basic operations, the biogeochemical cycles.
Okay, let's try to make sense of this metabolic mastery. How do we categorize it? Maybe by what they eat. Carbon source.
That's a great way to start. Based on their carbon source, we can divide them into two broad groups: autotrophs and heterotrophs.
Okay, autotrophs first.
Autotrophs are the self-feeders. They can build their own organic compounds from inorganic carbon, usually CO2. Within autotrophs, you have photoautotrophs, which use light energy to do this.
Like plants.
Exactly like plants in the case of cyanobacteria. They perform oxygenic photosynthesis using water, sunlight, and CO2 and releasing oxygen. A truly revolutionary invention.
Game changer for the planet.
Absolutely. But they're also anoxygenic photoautotrophs, green and purple sulfur bacteria, for instance. They use light energy too, but they use compounds like hydrogen sulfide, H2S, instead of water as their electron donor, so they don't produce oxygen.
Okay, different flavors of photosynthesis. What other autotrophs are there?
Then you have the chemolithoautotrophs. We mentioned them earlier, the rock eaters. They get their energy by oxidizing inorganic compounds, things like ammonia, nitrite, sulfur, iron, and they use that chemical energy to fix CO2 into organic matter.
So living off chemical energy, not light.
Right. Things like nitrifying bacteria in the soil are key examples. Then on the other side, you have the heterotrophs.
The ones that eat others?
Essentially, yes. They need to consume pre-existing organic compounds made by other organisms to get their carbon. Most bacteria we're familiar with, like E. coli in our gut, are chemorganoheterotrophs. They get both their energy and their carbon from organic molecules.
Okay, that seems straightforward.
There are also photoorganoheterotrophs, though they're less common. They use light for energy, but still need to consume organic compounds for their carbon building blocks. Some Rhodobacter species do this.
Light for power, organic stuff for parts.
You got it. And just to make it even more complex, some bacteria are mixotrophs. They're metabolically flexible and can switch between different modes depending on what's available. Autotrophy sometimes, heterotrophy other times.
That's incredible flexibility.
Okay.
So that's carbon source. What about energy source?
We can also categorize them based on energy source. Phototrophs use light, as we said. Chemotrophs use chemical energy.
Right.
And within chemotrophs, we distinguish between lithotrophs, which use inorganic chemicals as their electron donors, like those chemolithoautotrophs.
The rock eaters again?
Yep. and organotrophs, which use organic chemicals as their electron donors, like the chemorganoheterotrophs.
Okay, so multiple ways to slice the metabolic pie.
Hmm.
What are the core biochemical engines driving all this? The main pathways?
Several core pathways are fundamental across many bacteria and even other life forms. Glycolysis is a big one, the initial breakdown of glucose sugar into pyruvate. It yields a bit of ATP and some energy carriers like NADH. Very common.
Starting point for getting energy from sugar.
Right. Then, if oxygen is available, many bacteria use aerobic respiration. Pyruvate enters the citric acid cycle, or Krebs cycle, where it's further broken down, releasing CO2 and generating more energy carriers, NADH, and another one called FADH2.
More energy extraction.
Exactly. Then these carriers, NADH and FADH2, deliver their electrons to the electron transport chain. As electrons move down the chain, protons are pumped across the membrane, creating that proton motive force we talked about.
That electrochemical gradient.
Yes. And finally, that force is used to drive ATP synthesis, making lots of ATP. In aerobic respiration, oxygen is the final molecule that accepts the electrons at the end of the chain.
The reason we breathe oxygen.
Precisely. But, and this is crucial for bacteria, many can perform anaerobic respiration when oxygen isn't around. They use a similar process, but use other molecules as the final electron acceptor, like nitrate, sulfate, or even iron.
So they can still respire without oxygen.
Yes, though it generally yields less energy than using oxygen.
Yeah.
And then there's fermentation. This is another anaerobic process, but it doesn't use an electron transport chain at all.
How does that work?
It primarily involves glycolysis to make a little ATP, and then further reactions happen mainly to regenerate a key molecule called NAD plus PAP, which is needed for glycolysis to continue. It produces much less ATP than respiration, but it's vital in oxygen-free environments. Think about yogurt production or brewing that's bacterial or yeast fermentation at work. It was probably essential for early life in the oxygen-poor world.
Makes sense. And how do the autotrophs, the self-feeders, actually grab CO2 and turn it into organic molecules, the fixation part?
Right, CO2 fixation. There are actually several different biochemical pathways bacteria use to do this. The most famous and widespread is the Calvin cycle.
The one plants use.
Exactly. Used by cyanobacteria, purple bacteria, algae plants, and also many chemolithoautotrophs. The key enzyme here is rubisco, possibly the most abundant protein on Earth.
Wow. Any other ways?
Yes. Some anaerobic bacteria, like the green sulfur bacteria, use a pathway called the reverse TCA cycle, basically running the citric acid cycle backwards to build molecules instead of breaking them down. And still others, like certain anoxygenic phototrophs, chloroflexus, and some archaea, use pathways like the hydroxypropionate pathway.
Multiple solutions to the same problem.
It really suggests either that this ability evolved independently multiple times because the pressure to be autotrophic on early Earth was so strong, or perhaps these pathways diversified from an ancient common ancestor. Either way, it highlights incredible evolutionary innovation.
It's just astonishing, this metabolic toolkit they have. It explains so much about why they dominate almost every environment.
Exactly. Their metabolic versatility is absolutely key to their ecological success, their resilience, and their indispensable roles in the global biogeochemical cycles that sustain all life. They're the planet's master chemists.
Okay, so we have these incredibly diverse bacteria thriving metabolically. How do they actually reproduce? And maybe more importantly for evolution, how do they change and adapt so effectively over time?
Well, the primary way bacteria reproduce is through binary fission. It's a form of asexual reproduction. Basically, the cell grows, replicates its single chromosome, and then divides right down the middle into two genetically identical daughter cells.
Identical copies, so fast multiplication.
Very fast under the right conditions. Some bacteria can divide every 20 minutes. This allows for rapid population growth. But, as you noted, because the daughters are identical clones, binary fission itself doesn't generate much genetic diversity within that lineage.
Right. Good for numbers, not so much for new ideas or adaptations on its own. So where does the variation come from?
This is where horizontal gene transfer HGT becomes incredibly important. Unlike vertical inheritance from parent to offspring, HGT is the transfer of genetic material between existing individual organisms. It's a major, major driver of bacterial evolution.
Okay, HGT. How does that actually happen?
There are three main mechanisms we know of. First, there's transformation. This is when a bacterium takes up naked DNA directly from its environment. This DNA might have been released from other bacteria that have died and broken open or released.
They just slurp up loose DNA.
Kind of. But the recipient cell usually needs to be in a specific physiological state called competence to be able to do this. Once the DNA is inside, it can either be integrated into the bacterium's own chromosome through recombination, or sometimes, if it's a plasmid, it can just exist and replicate separately. Fun fact. Transformation was actually the process used in the famous Avery McLeod-McCarty experiment that proved DNA carried genetic information.
Wow. Okay. What's the second way?
Second is transduction. This involves bacteriophages, viruses that specifically infect bacteria.
Viruses moving bacterial genes around.
Exactly. During the phage's replication cycle inside a bacterium, sometimes a piece of the bacterial DNA gets mistakenly packaged into a new virus particle instead of, or along with, the viral DNA. When that phage then infects another bacterium, it injects that piece of bacterial DNA from the previous host.
Like a little genetic delivery service.
Pretty much. This can be generalized, meaning potentially any bacterial gene can get transferred by accident or specialized, where only genes located near the specific site where the phage DNA integrates into the bacterial chromosome are transferred frequently.
Okay, transformation, transduction. What's the third one?
The third is conjugation. This is often described as bacterial mating, but again, it's not reproduction. It involves direct cell-to-cell contact.
They actually touch.
Yes. It's usually mediated by special plasmids called conjugative plasmids, like the famous F plasmid in E. coli. These plasmids carry genes that allow the donor cell to produce a structure called a sex pilus.
The sex pilus we mentioned earlier.
That's the one. The pilus forms a bridge between the donor and recipient cell. Then a copy of the plasmid, and sometimes even parts of the donor's main chromosome that are attached to it, can be transferred across this bridge into the recipient cell.
So direct injection of DNA through a tube. That sounds very efficient.
It can be very efficient for transferring plasmids, yeah.
So transformation, transduction, conjugation.
Yeah.
3 ways to share genes sideways. What's the big picture significance of all this HGT?
The significance is just immense for bacterial evolution. HGT allows bacteria to acquire novel genes and traits incredibly rapidly. Think about gaining antibiotic resistance, or new metabolic capabilities, or factors that help them cause disease, virulence factors. They don't have to wait for slow, random mutations to maybe create these traits. They can potentially just borrow them from their neighbors.
A genetic shortcut.
Exactly. The acquired DNA can then be stably integrated into the recipient's genome, usually through a process called homologous recombination, if it shares similarity with existing DNA. This constant interplay between rapid multiplication via binary fission, which quickly spreads any beneficial trait within a population, and HGT, which introduces entirely new genetic possibilities from outside, creates an incredibly potent evolutionary dynamic.
Different from how we eukaryotes mostly evolve, right?
Very different from the predominantly vertical inheritance, parent to offspring, and sexual recombination that drives evolution in most animals and plants. HGT makes bacterial genomes remarkably fluid and modular. It's almost like there's a shared genetic commons where useful adaptations can spread quickly across different species, even distantly related ones.
And we see the consequences of that today.
Absolutely. The alarming speed at which antibiotic resistance spreads through bacterial populations globally is a direct and very concerning consequence of HGT in action. It's evolution happening on a timescale we can observe.
Right. A biological internet constantly sharing updates. Sometimes updates we really don't like. Okay, so bacteria are established, diverse, reproducing fast and swapping genes. Now let's get to the really big planetary shifts they caused, the Great Oxidation Event.
Yes, the GOE, the Great Oxidation Event. This is arguably one of the most profound transformations our planet has ever undergone, and it was driven entirely by bacteria.
Which bacteria specifically?
Specifically cyanobacteria. These were the innovators that evolved oxygenic photosynthesis, probably somewhere around 3.0 to 2.7 billion years ago. They figured out how to use sunlight to split water molecules, H2O, to get electrons, releasing oxygen, O2, as a waste product.
And before this, virtually no free oxygen in the atmosphere or oceans.
That's the picture, yes. Any free oxygen produced by, say, UV light splitting water vapor would have been immediately mopped up by reacting with reduced minerals on the surface and dissolved in the oceans, especially iron.
So the oxygen didn't build up.
Not at first. As cyanobacteria started pumping out oxygen, it reacted with all that dissolved iron in the oceans, causing it to precipitate out as iron oxides. This is what formed those massive geological formations we call banded iron formations, or BIFs. They're basically ancient rust layers in the rock record, a key piece of evidence for the GOE.
So the oceans literally rusted.
In a manner of speaking, yes. Eventually, over hundreds of millions of years, these oceanic oxygen sinks, primarily the dissolved iron, became saturated. There wasn't much left for the oxygen to react with in the water.
And then it started escaping into the atmosphere.
Exactly. Around maybe 2.4 to 2.2 billion years ago, oxygen levels began to rise significantly in the atmosphere and surface oceans.
That must have been, well, revolutionary doesn't seem strong enough. What did that do to the existing life?
For many of the anaerobic organisms that had dominated the planet up to that point, it was an absolute catastrophe. Oxygen was highly reactive and toxic to their metabolisms.
Poison.
A poison, yes. This likely triggered mass extinction events. Many anaerobic lineages probably died out completely, while others were forced to retreat into oxygen-free environments, deep sediments, hydrothermal vents, places like that. Those became anoxic refugia.
So, in a sense, this was Earth's first major pollution crisis, caused by life itself.
That's a perfect way to put it, entirely biological pollution. But here's the incredible paradox. This pollution event, this oxygenation, fundamentally reshaped the planet's chemistry and, crucially, created the conditions necessary for a whole new type of life to evolve: aerobic life.
Life that could use the oxygen.
Exactly. Organisms that evolved aerobic respiration could harness the powerful oxidizing potential of oxygen to extract far more energy from their food than anaerobic organisms could. This opened up entirely new evolutionary possibilities, eventually leading to larger, more complex cells.
So the air we breathe, the foundation for us and all animals, is basically leftover waste from ancient bacteria. That's humbling.
It really is a byproduct that changed the world.
And I think you mentioned earlier, there's some hint that maybe some bacteria could handle oxygen even before the GOE really got going.
Yes, that's a fascinating wrinkle. Some phylogenetic studies and analyses of metabolic pathways suggest that certain bacterial lineages might have evolved the machinery to deal with and maybe even use small amounts of oxygen, perhaps hundreds of millions of years before oxygen levels shot up globally.
How could that be?
Well, maybe there were localized oxygen oases near cyanobacterial mats even before the atmosphere filled up. Or perhaps enzymes originally used for detoxifying reactive oxygen species were gradually co-opted for respiration. There's evidence suggesting an ancestor of cyanobacteria themselves might have been among the earliest to figure out how to use oxygen metabolically.
So evolution was maybe tinkering with oxygen use even before it became widespread.
It seems plausible. It's a good reminder that evolution is often messy and opportunistic, sometimes developing solutions slightly ahead of the main environmental shift.
Okay, so oxygen is rising, anaerobes are retreating or dying off, and new aerobic bacteria are starting to thrive and diversify. What happens next in this bacterial saga?
Well, the rise of oxygen really fueled further diversification. Those lineages that could adapt to use oxygen gained a huge energetic advantage. We see the ancestors of many of the major bacterial phyla that exist today appearing in the fossil record and in phylogenetic reconstructions during this period, roughly between 2.5 and 1.8 billion years ago. This really established a long era of bacterial dominance.
So the major groups we know today have roots going way back.
Deep roots, yes. While the earliest lineages were mostly anaerobic, the ability to survive and thrive aerobically seems to have evolved independently several times as oxygen levels rose. But anoxygenic photosynthesis also remained a very successful strategy, and it's still found in about half of the major bacterial lineages today.
It started colonizing more places.
Absolutely. Bacteria pushed into a wider range of environments, including really extreme ones like deep-sea hydrothermal vents back in the Precambrian, and remarkably, some relatively primitive modern bacteria found in those vents today seem to be descendants of those early colonists. It was this constant dance of coevolution. Bacterial metabolism, like the cyanobacteria producing oxygen or methanogens producing methane, potentially warming the early climate, changed the Earth's geochemistry.
And those geochemical changes then drove further bacterial evolution.
Exactly. It created new pressures and new opportunities, like the evolution of efficient aerobic respiration in response to the very oxygen the cyanobacteria produced.
This intricate feedback loop, and all of this deep bacterial history, this metabolic innovation, this planetary engineering, it sets the stage for the next truly enormous leap, doesn't it? The origin of complex cells, the eukaryotes.
Precisely. This is where bacteria play another absolutely central transformative role. The transition from those relatively simple prokaryotic cells, bacteria and archaea, to the much larger, more complex eukaryotic cells. That's us. Plants, fungi, protists. That happened somewhere around, let's say, 1.6 to 2.2 billion years ago. It was a quantum leap in biological organization.
And the key idea here is endosymbiosis, right?
Endosymbiosis is the cornerstone of our understanding, yes.
Yes.
The theory posits that eukaryotic cells arose from symbiotic relationships where one prokaryotic cell engulfed another, and instead of digesting it, they formed a permanent partnership.
The cell within a cell idea. Let's talk about the most famous example, mitochondria. Where did our cellular power plants come from?
Mitochondria, which are essential for efficient energy production through aerobic respiration in almost all eukaryotes, are now overwhelmingly understood to have originated from an aerobic bacterium, specifically an alpha proteobacterium.
An alpha proteobacterium.
Okay. This bacterium was engulfed by a host cell, which was likely an archaeon, probably one related to those Asgard archaea we'll get to.
So an archaeon ate a bacterium.
Engulfed, yes. And this likely started as a mutualism, a beneficial relationship. The engulfed bacterium was great at making ATP using oxygen, which benefited the host. And the host provided protection and maybe a steady supply of nutrients or fuel to the bacterium.
A win-win.
Initially, perhaps. But over vast stretches of evolutionary time, this engulfed bacterium became increasingly integrated into the host cell. It lost many of its own genes, transferring some essential functions to the host genome. It basically became domesticated, evolving into the mitochondrion we know today, retaining only a small remnant of its own DNA and its own bacterial-style ribosomes.
Wow, so we literally have the descendants of ancient bacteria living inside almost every one of our cells.
That's exactly right. And then for plants and algae, there was another major engulfment event later on, right?
Correct. After the evolution of the mitochondrion-containing eukaryotic cell, one lineage of these early eukaryotes went on to engulf another bacterium this time, a photosynthetic cyanobacterium, the ones that cause the GOE.
The very same group. This second endosymbiotic event gave rise to chloroplasts, the organelles that perform photosynthesis in plants and algae. And interestingly, the evidence suggests that primary chloroplast endosymbiosis might have happened just once, but secondary and even tertiary engulfments, where a eukaryote engulfs another eukaryote that already has chloroplasts, seem to happen multiple times independently in different algal lineages. It gets complicated.
It really does sound like evolution by committee, or maybe evolution by acquisition. What's the evidence supporting this whole endosymbiotic story? It sounds pretty wild.
It does sound wild, but the evidence is incredibly strong and comes from many different lines. First, both mitochondria and chloroplasts have two membranes surrounding them. The inner membrane has characteristics similar to a bacterial membrane, likely derived from the original symbiont, while the outer membrane resembles the host cell's membrane, likely formed during the engulfment process.
Okay, double wrapping.
Then they have their own DNA. It's typically a small circular chromosome, just like in most bacteria, and distinct from the linear chromosomes in the eukaryotic nucleus.
Their own little instruction manual.
Exactly. They also have their own ribosomes, the machinery for making proteins based on that DNA. And these ribosomes are the 70S type, the bacterial size, different from the 80S ribosomes in the eukaryotic cytoplasm.
Just like you mentioned with antibiotics.
Right, it fits perfectly. Furthermore, mitochondria and chloroplasts reproduce by binary fission, dividing independently within the host cell, much like free-living bacteria do.
They make copies of themselves inside our cells.
They do, and perhaps the most compelling evidence comes from genetics. Phylogenetic analyses comparing the DNA sequences from mitochondria and chloroplasts with those of modern prokaryotes consistently show that mitochondrial DNA is most closely related to alpha proteobacteria, and chloroplast DNA is most closely related to cyanobacteria. The genetic footprint is undeniable.
The DNA evidence nails it.
Pretty much. And finally, there's the phenomenon of endosymbiotic gene transfer. Over time, many genes originally present in the genomes of the engulfed bacteria have actually moved into the host cell's nucleus. The proteins encoded by these genes are then synthesized in the cytoplasm and imported back into the mitochondrion or chloroplast. This creates an irreversible dependency.
So the organelle can't survive on its own anymore, and the host relies on it. They're truly integrated.
Exactly. It solidifies the merger. There's this idea of an HGT ratchet gene transfer tends to go from the organelle to the nucleus, making it harder and harder to reverse the process. It locks them together.
Okay, so this incredible merger happens, involving an archaeal host and a bacterial symbiont. This gives rise to the first eukaryotes. What do we know about the common ancestor of all living eukaryotes, LSE? The last eukaryotic common ancestor. Was it simple or already quite complex?
That's a key question. And the picture emerging of LSE is one of surprising complexity. It wasn't some simple intermediate. LSCA likely already possessed mitochondria, or at least organelles derived from that initial endosymbiosis.
So mitochondria were there from the start of eukaryotes as we know them.
It seems very likely, yes. LACA also almost certainly had a nucleus containing linear chromosomes, an endomembrane system, including structures like the endoplasmic reticulum and Golgi apparatus, which handle protein and lipid processing and transport, and a cytoskeleton made of proteins like actin and tubulin, providing internal structure and motility.
Wow, that sounds pretty advanced already.
It does, and it probably had sophisticated genetic systems too, a larger genome than prokaryotes, introns, those non-coding sequences within genes that need to be spliced out, probably RNA interference mechanisms for gene regulation, complex cell division processes, and maybe even the ability to engulf particles through phagocytosis.
Mm.
Metabolically, it was likely a mosaic, capable of aerobic respiration thanks to its mitochondria, but possibly retaining some anaerobic capabilities inherited from its archaeal host and maybe other bacterial gene transfers.
So LACA wasn't a gradual step up. It seems like a lot of eukaryotic complexity arrived in a burst almost.
That's what the evidence increasingly suggests. A relatively rapid burst of innovation seems to have occurred around the time of, or shortly after, the acquisition of mitochondria. It's like the energy boost from mitochondria enabled or facilitated the evolution of all these other complex features quite quickly. Lessier appears almost ready-made in many respects.
And this reinforces the idea of eukaryotes being fundamentally chimeric, a blend of origins.
Absolutely. Lessii was clearly a chimera. Its core information processing machinery, DNA replication, transcription, translation seems largely derived from its archaeal ancestor. Its energy metabolism was revolutionized by the alpha proteobacterial endosymbiont, and other cellular functions likely drew on contributions from various other bacterial and archaeal sources through HGT. Eukaryotic evolution wasn't just a simple merger, it was weaving a complex tapestry from different evolutionary threads.
Okay, let's talk more about that archaeal host. You mentioned Asgard Archaea. This has been a huge discovery, right?
Huge. The discovery and study of the Asgard superphylum of Archaea groups, named after Norse gods like Loki Archaeota, Thor Archaeota, Odin Archaeota, Heimdallr Iota, has been truly revolutionary for understanding eukaryotic origins.
Why? What's special about them?
Because genetic analyses show they are, by far, the closest known prokaryotic relatives to eukaryotes found so far. They branch closer to eukaryotes on the tree of life than any other archaea or bacteria.
Closer relatives than other archaea.
Yes, and what's really exciting is that their genomes contain genes for proteins that were previously thought to be unique to eukaryotes. These are often called eukaryote signature proteins, or ESPs.
What kinds of proteins are these?
They're involved in functions that are hallmarks of eukaryotic cell complexity, things like components of the cytoskeleton, proteins involved in vesicular trafficking, moving things around inside the cell in membrane bubbles, parts of the ubiquitin system, which tags proteins for degradation, and proteins involved in remodeling cell membranes.
So, the archaeal ancestor wasn't just a simple blob. It already had some of the genetic toolkit for building a complex eukaryotic cell.
That's exactly what it suggests. The archaeal host that engulfed the protomitochondrion might have already been somewhat complex itself, perhaps possessing a rudimentary cytoskeleton and internal membrane system. This might have primed it, making it more capable of engulfing another cell in the first place. It seems the Asgard lineage contributed key components to many fundamental eukaryotic systems, well beyond just the basic information processing.
That really changes the picture of the host.
It does, and studying the metabolism of modern Asgard archaea, which are often found in deep-sea sediments and live in syntrophy with other microbes, is also giving us clues about the potential metabolic interactions that might have occurred during the initial symbiosis. For instance, maybe there was an exchange of hydrogen or electrons between the archaeal host and the bacterial symbiont.
And this close relationship has implications for the whole tree of life structure.
Absolutely. The discovery of Asgard archaea strongly supports the idea that eukaryotes didn't just branch off alongside archaea as their domain, but actually emerged from within the archaeal domain. This has led to concepts like eukaryomorpha, grouping Asgard archaea and eukaryotes together, effectively making eukarya a branch within archaea and challenging the traditional three-domain tree: bacteria, archaea, eukarya that we often see.
Wow, so it might be more like two primary domains with eukaryotes nested inside one of them. Fascinating. Now, endosymbiosis is clearly the central theory, especially with the Asgard discoveries. Are there any other ideas or complementary models about how eukaryotes arose that are still considered?
Yes, while endosymbiosis involving an Asgard-like archaeon and an alpha proteobacterium is the leading model, there are other hypotheses that try to explain specific features of eukaryotes or offer different perspectives.
Like what?
Well, there's the viral eukaryogenesis, VE hypothesis. This suggests that the nucleus itself might have originated from a large, complex DNA virus that infected an archaeal host.
A virus became the nucleus.
That's the idea. Proponents argue it could potentially explain features like the nuclear membrane, derived from the viral capsid or host membranes, the linear chromosomes typical of eukaryotes, many viruses have linear DNA, and even some unique nuclear processes like mRNA capping and the separation of transcription in the nucleus and translation in the cytoplasm.
Provocative. What else?
There's the inside-out model. This offers a different scenario for the physical process. It proposes that the archaeal ancestor might have extruded large membrane protrusions or blebs that surrounded external symbiotic bacteria, the protomitochondria. The cytoplasm would then have formed from the fusion of these blebs, with the original cell body becoming the nucleus.
So the cytoplasm formed around the symbionts, rather than engulfing them inwards.
Kind of, yes. It flips the perspective. And then there's the older eocyte hypothesis, which originally proposed that eukaryotes arose from within a specific group of archaea called Crenarchaeota, or Thermoproteota, sometimes called eocytes. This idea actually predates the Asgard discovery, but has been somewhat revitalized by it, as both place eukaryotic origins firmly within the archaea, even if they point to slightly different archaeal ancestors.
So these aren't necessarily mutually exclusive with endosymbiosis.
Not always. Some aspects, like the viral origin of the nucleus, could potentially be layered onto an endosymbiotic framework. The inside-out model is primarily about the physical mechanism of engulfment or surrounding. They often try to address specific puzzles that the standard model might not fully explain on its own. It's still a very active area, and researchers are working hard to integrate all the genomic and cell biological data to get the most complete picture.
It's clear bacteria were fundamentally involved in getting eukaryotes started. But their influence didn't just stop there, right? They've kept shaping eukaryotic evolution ever since.
Absolutely not. The initial endosymbiosis was just the beginning of a very long and ongoing relationship. Bacteria have continued to exert a profound influence on eukaryotic evolution through mechanisms like continued horizontal gene transfer and by fundamentally shaping the environments where eukaryotes live and evolve.
So even after eukaryotes got their mitochondria and chloroplasts, they were still picking up genes from bacteria.
Yes, indeed. While maybe not as rampant as HGT between prokaryotes, HGT from bacteria to eukaryotes has definitely occurred and has had significant impacts, especially in single-celled eukaryotes, the protists, but also leading to specific adaptations in multicellular life.
How does that happen? How do eukaryotic cells pick up bacterial genes?
Well, there's the ongoing endosymbiotic gene transfer. We talked about genes continuing to move from mitochondria and chloroplasts to the nucleus, but also phagocytosis, the process many protists use to eat bacteria, can inadvertently lead to the uptake and potential integration of bacterial DNA.
Eating your neighbor and stealing their genes.
Something like that. Other potential routes include transformation, less common in eukaryotes but possible, perhaps viral vectors carrying bacterial genes, or through very intimate long-term symbiotic relationships between eukaryotes and bacteria. That same HGT ratchet might apply once a gene gets into the nucleus and becomes functional. It's hard to lose.
So what kinds of traits have eukaryotes actually gained from bacteria this way after the initial big mergers?
A real variety of things, often related to metabolism or adapting to specific niches. We see many examples of metabolic enzymes being transferred. For instance, many anaerobic protists living in oxygen-poor environments have acquired genes for fermentation pathways from bacteria. Some dinoflagellates seem to have picked up bacterial genes that help them cope with losing certain mitochondrial functions. Fungi also have a number of metabolic genes that appear to be bacterial in origin.
So boosting their metabolic toolkit.
Exactly. HGT has also helped eukaryotes adapt to new lifestyles. Think about plant parasitic nematodes, or some insects. They've acquired genes for enzymes that break down plant cell walls, likely from bacteria, allowing them to feed more effectively.
That makes sense. Any other examples?
Sure. There's evidence for acquired stress responses. For example, tardigrades, those incredibly resilient microscopic animals, seem to have acquired a gene involved in radiation tolerance from bacteria. Some marine animals, like hydrozoans, appear to have gotten genes for producing protective antioxidant compounds called ovoofuels from symbiotic bacteria living with them.
Wow, borrowing survival tricks.
Right, and even things like nutritional supplementation. In many insect bacterial symbioses, the bacteria provide essential amino acids that the insect can't make, and sometimes the genes for these pathways show signs of having been transferred to the host genome eventually.
So it's a real grab bag of useful genetic tools.
Yeah.
It must be tricky for those genes to work in a eukaryotic cell, though.
It is. For a transferred gene to function, it needs to successfully integrate into a chromosome, acquire the right regulatory sequences, promoters, etc., so it gets turned on and off correctly in the eukaryotic context. And if the protein needs to go to a specific place, like an organelle, it might need to gain a targeting signal. Sometimes acquiring introns can even help stabilize or enhance its expression in the eukaryotic system. But despite these hurdles, it clearly happens often enough to be evolutionarily significant. HGT has provided eukaryotes with a sort of genetic library to borrow from for rapid adaptation, especially for metabolism and exploiting new resources.
And like you said, it's ironic that phagocytosis, a key way eukaryotes eat, also opens the door for this gene theft.
Exactly. It creates this potential evolutionary feedback loop. Eating bacteria provides food, but also occasional genetic upgrades.
Okay, so direct gene transfer is one major ongoing influence. How else have bacteria shaped eukaryotes after their origin?
Through their sheer ecological dominance and metabolic activity, bacteria have fundamentally shaped the physical and chemical conditions of almost every environment on Earth where early eukaryotes evolved and diversified.
Shaping the stage on which eukaryotic evolution played out.
Precisely. Bacteria drive the major biogeochemical cycles: nitrogen, sulfur, carbon, etc. They dictate nutrient availability, the redox state of the environment, how much oxygen or other oxidizing reducing agents are around, and the presence of countless specific chemical compounds. Early eukaryotes didn't evolve in a vacuum. They evolved within these complex, bacterially dominated ecosystems. Their own metabolism, their physiology, was inevitably shaped by and adapted to the chemical landscapes created by bacteria.
Can you give an example?
Sure, think about sulfur cycling. Bacteria play the main roles in oxidizing and reducing sulfur compounds. Early eukaryotes evolving in environments with active sulfur cycling likely had their own sulfur metabolism pathways influenced or even acquired from these bacterial neighbors.
Okay. What about oxygen levels? We know bacteria caused the GOE, but levels fluctuated after that, right?
They did. And crucially, even after the GOE, many environments likely remained low in oxygen or fluctuated, partly due to the activity of anaerobic bacteria consuming oxygen or producing reducing compounds. Eukaryotes likely originated and did much of their early diversification in such low oxygen or fluctuating oxygen conditions.
So they needed to cope with low oxygen from the start.
It seems very likely. And we see evidence for this in the genomes of many modern eukaryotes, especially early diverging anaerobic protists. They possess enzymes and pathways for anaerobic energy metabolism, things like PFO, pyruvate.ferredoxin, oxaloreductase, and phe-hydrogenases, which are typically found in anaerobic bacteria and archaea.
Yeah.
Their widespread distribution suggests they were present in early eukaryotic ancestors.
So even though mitochondria gave them aerobic power, they kept their anaerobic toolkit.
Yes, it suggests that being able to function both with and without oxygen, being facultatively anaerobic, might have been the ancestral state for eukaryotes, allowing them to thrive in those variable early environments shaped by bacteria. The ability to perform efficient aerobic respiration was a huge advantage when oxygen was plentiful, but retaining anaerobic options was probably critical for survival.
And intimate symbioses continued to be important too, beyond just the big organelle ones.
Absolutely. Close physical associations, including syntrophic relationships where different microbes live off each other's metabolic byproducts, were likely common, especially in anoxic environments. These interactions would have facilitated the exchange of metabolites and, as we discussed, potentially genes, further shaping eukaryotic metabolism. That ovothile biosynthesis example likely arose from such a close symbiosis before the genes were transferred.
So it really hammers home the point. Bacterial metabolism actively sculpted the planetary environments, setting the stage, defining the challenges, the niches, the opportunities, basically writing the rules of the game for early eukaryotic evolution.
You've got it. The prevalence of those bacterial-like anaerobic pathways in many diverse eukaryotes is strong evidence that adapting to the world defined by bacteria, particularly low oxygen conditions, was fundamental from the very beginning of eukaryotic history.
So it's this incredibly deep, ongoing, intertwined story. Bacteria didn't just kickstart eukaryotic complexity. They've been constant partners, influencers, and environmental engineers throughout the entire history of eukaryotic life.
Precisely. You really can't understand the evolution of eukaryotes without understanding the bacteria that came before them and continue to surround them. Their evolutionary destinies are, and always have been, profoundly linked.
So, to kind of wrap this up, we've taken quite a journey. From those very first bacterial pioneers on a harsh young Earth, through the planet-altering great oxidation event driven by cyanobacteria, then the incredible symbiotic mergers endosymbiosis that gave rise to the complex eukaryotic cell.
A merger likely involving an Asgard-related archaeon and an alpha proteobacterium.
Right, leading to Lithier, that surprisingly complex ancestor. And finally, realizing that bacteria didn't just fade into the background. They've continued to shape eukaryotes right up to the present day through ongoing gene transfer and by constantly defining the environmental context.
It really highlights that evolution isn't always this neat linear progression. It's often contingent, opportunistic, driven by these incredible partnerships, symbioses, and the ability to share innovations horizontally.
Yeah, it definitely challenges that simple branching tree of life picture we often see. It sounds much more like an interconnected web, or maybe that ring of life idea, especially near the base, showing how deeply archaea and eukarya are related.
Exactly. And it emphasizes that biological complexity, the amazing features of eukaryotic cells, didn't necessarily just arise from slow, gradual accumulation within one lineage. Often it seems to spring from these synergistic combinations, putting together different parts from different origins to create something fundamentally new.
It really makes you think about how much more there is to discover. Exploring that vast microbial dark matter, all the bacteria and archaea we haven't even cultured or studied yet, figuring out the precise step-by-step mechanisms of eukaryogenesis. Understanding the full extent and impact of all this horizontal gene transfer throughout history. Reconstructing what those ancient ancestors really looked like.
Absolutely. Those are all huge and incredibly exciting frontiers. Developing new tools, better sequencing, better bioinformatics, maybe even cultivating some of these elusive Asgard archaea will continue to shed light on these fundamental questions about the history of life.
So for you, our listener, here's a final thought to maybe chew on. That very complexity we associate with advanced life, even with ourselves,
Yeah.
it might not primarily be a story of solitary lineages driving upwards. Instead, maybe it's a far more intricate, dynamic, and collaborative story. A story of ancient partnerships, ongoing conversations, and shared innovations forged billions of years ago at the microbial level and continuing even now.
A history built on connection.
It's exactly a history of life constantly borrowing, merging, and evolving together in ways we're only just beginning to fully appreciate. We really encourage you to keep exploring this dynamic and deeply interconnected story of life on our planet. Thanks for taking the deep dive with us.